This is Selma Schimmel in Chicago at the annual ASCO meeting for the group room. Happy to be joined by Dr. David Quinn, Medical Director of the Norris Cancer Hospital, co-leader of the Genitourinary Cancers Program for the USC Norris Comprehensive Cancer Center, head of the section of Genitourinary Medical Oncology, Associate Professor of Medicine in the Division of Cancer, Medicine, and Blood Diseases at the Keck School of Medicine at USC, which is in Southern California, our hometown.
Good afternoon, Selma. How are you?
How are you, Dr. Quinn?
I'm very well, thank you.
We so look forward to our annual meetings with you. One of the areas I would like to get caught up about is what's new in bladder cancer.
It's been a quiet meeting at ASCO related to bladder cancer, and GU ASCO is fairly quiet as well. A lot of research activity, but not much progress. In fact, we had a commentary here at ASCO on bladder cancer that made the initial statement that we probably hadn't made too much progress in the last 25 years, especially related to advanced disease. And that's fairly depressing given the progress we've made in some of the other cancers in that time.
What sort of things are on the horizon and what's currently being researched?
The horizon falls into a number of areas. We're trying to understand different types of bladder cancer based on the molecules that are abnormal inside the cancer. Some of these things are called oncogenes. They're specifically cancer genes that are switched on, or tumor suppressor genes. They're genes in cancer that are switched off. And we've known about these for quite some time in bladder cancer. What we're looking for to move forward with a therapy is we want something called targetable molecules, targetable oncogenes, targetable tumor suppressor genes, or other molecules that are present in the cancer.
Are there any clinical trials or drugs in development that patients should know about?
One of the things that I think is most interesting is a new molecule that's been designed to inhibit something called heat shock protein 27. And we have a variety of heat shock proteins in our body, and the reason we give them different numbers, so we have heat shock protein 90 and heat shock protein 70, is they have different molecular weights. So this one happens to have a molecular weight of 27. And the heat shock proteins have a variety of functions in the body, most particularly regulating cells in response to stress. So if we stress a normal cell, we want the heat shock proteins to switch on to protect it. There's a lot of redundancy amongst the heat shock proteins. Heat shock protein 27 seems to be very important in certain cancer cells for maintaining survival in the face of chemotherapy, but also in the face of other insults like radiation, for example. And is also, if there's too much of it, tends to be a survival factor that will mean the cancer cells grow more rapidly. So what we've seen developed in the laboratories of Oncogenics, a company based in Vancouver, British Columbia, is an antisense compound that can be given intravenously once a week that inhibits heat shock protein 27, and that's called OGX-427. And we've moved that into trials in a variety of areas. Now, in bladder cancer, historically, targeted therapies have usually either worked in superficial low-grade disease or in advanced disease. The interesting thing about this therapeutic in the work that has been done so far is that it can be placed in the bladder for patients with lower grade superficial disease and seems to have very significant activity. What's also interesting and sort of closer to where I deal with bladder cancer, which is more when it's become more aggressive, is that if we administer this compound, it does appear to have some activity on its own against bladder cancer. It appears to be tolerable even though you've got to have it every week. It's a fairly simple IV, it's a peripheral IV, you don't have to have a central line. It tends to produce some shakes and chills in patients similar to what we see with Taxol or with rituximab, some of the monoclonal antibodies. Interestingly, significant activity in combination with chemotherapy in preclinical models, and some early indications from early studies that it could potentiate chemotherapy and maybe even be synergistic with chemotherapy when we co-administer it. So we're working on a trial at the moment for patients who have metastatic disease, and the design of the trial has 3 arms, and one of the arms, the patients get standard chemotherapy, and they get that at a standard dose for a standard length of time, just as they would otherwise. In the other 2 arms, we add an infusion on a weekly basis of the OGX 427. And patients are going to get 2 doses. And you might say, why are we giving 2 doses? And the answer is that we don't quite know yet exactly what the biological dose is to best help the chemotherapy kill the cancer. And so there will be some collection of particular cells to see how much the heat shock protein is inhibited, to see how well the chemotherapy goes. And we know that the doses given at the 2 different dose levels are tolerable with no real difference in side effects. So we're trying to get that scheduling right.
Dr. Quinn, this is compelling and hopeful. Patients that are listening and are interested in becoming involved in this study,
yes,
what do they have to do?
The study is on the clinicaltrials.gov website. It's going to be open broadly across North America, both in Canada and the United States.
When you go to the website, how do they search it? Do they put in bladder cancer trials?
It'll come up in bladder cancer trials, and you'll get a lot of hits. Unfortunately, when people do that, they find a lot of old trials that are no longer open. So what I tell my patients to do is if they want to get that information, put in 427, and they will find it.
Thank you so much.
Thank you, Selma.
Pleasure.